Heard about PCR biosystems?
There's quite a buzz about us.
Hop on over to our website to find out more.
Welcome to the Our Body session. I hope you are having a wonderful evening and you accompany us with your preferred pint. My name is Anna Yaramilo. I'm a PhD student of computer science at the University of Exeter, and I'll host this session. Today we will have 5 exciting presentations about navigating our existence with illnesses and diseases, and how better it is preventing than cure. The pandemic has permeated all aspects of our life, and we are all keen to leave COVID-19 behind. I have a question for you to think about during this session, and please write your answer in the chat box. The chat box is in the lower part of the video in the YouTube channel. The question is, what is the first thing, activity, travel you would do after receiving the COVID-19 vaccine and you are free to go anywhere? While you think about the question and you write your answers in the chat box, this session wants to sow a seed in your mind. It may be time to think about what other pandemics come next and the importance of being aware that diseases and illnesses have always existed, so we could start to better prepare for the future. Thinking about what we can do to better coexist with diseases and illnesses and how a more positive attitude can help us. Now let me talk about some house rules for this session. We will have 3 main speakers with 10-minute presentations about vaccines, diseases, and attitudes toward aging. Also, we will have 2 flash talks of 3 minutes about homeless women and augmented reality. If you have questions or comments during all the sessions, please write them in the chat box of the YouTube channel. In the final part of this session, we will have a 10 minutes Q&A session. In that last session, I'll show you some of the answers to our question about future plans for COVID-19 after the vaccine. And the speakers will discuss your questions. So please remember to post them at any time during the presentations in the chat box and also during the question session. Now, I'm very excited to present you our first speaker, Dr. Nic Harmer. He is a professor of biochemistry at the University of Exeter and an expert in the use of enzymes to produce vaccines. His talk today will be about discussing how vaccines help to give us immunity against infectious diseases. Nicholas, the stage is yours.
Thank you. Okay, so vaccines are one of humanity's greatest tools for improving public health. We now have vaccines against more than 25 of the world's most dangerous diseases. Before COVID-19, vaccines saved at least 2 million lives every year around the world. The success of the COVID-19 vaccines has shown many of us for the first time how important the vaccinations we've had in our lives are. However, we don't have vaccinations against all diseases. My question for the audience, and again, you can put this in the chat box or maybe have some of the answers come up on screen or be discussed later. If you had the choice to fund a vaccine against any one disease, which one would it be and why? Now, usually, as we've heard about it over the last year, vaccines often take 10 to 20 years to get from the laboratories where people start research to patients where they're going to get given to people in large numbers. And there's these long pipelines to get there. Vaccines against COVID-19, thankfully, have taken less than a year to be ready. Partly, this has been because there's been rapid investment by governments and companies. And that's meant that some of those stages like manufacturing, which otherwise take many years, have been invested in while the clinical trials are going on. And that's shaved years off the process to get the vaccines into humans. Mostly, though, it was because teams have built technologies that allowed them to get rapidly from the first results to having a vaccine candidate, and that these were able to rapidly adapt to the new threat. What I'm going to talk about briefly is why these work, and will they work against every threat that we may find in the future. Now, vaccines work by training our bodies to respond to infection threats. The way our bodies do this is that they continuously produce cells that act as sentinels to go around our bodies and look for infectious agents like bacteria and viruses. When they find suspect molecules, these are used as a template to activate two other types of cells that act as the defense for our bodies. One of these is the B cells here, which produce antibodies. And these antibodies are released into the bloodstream, and they then tag invaders for destruction. The other type of cells are called T cells. And what these do is they recognize templates that indicate an infected cell in our body. And once they're activated, the T cells move into the bloodstream and act essentially as Death Stars, seeking out infected cells and eliminating them so that those infected cells can't go on to cause more infections. A good vaccine will help our bodies to produce both antibodies and T cells. And the vaccines being developed today achieve this by taking the best parts of the invader that will produce the best immune response. And they present only that part to the immune system, not a whole bacteria with lots of decoys that won't make such a good response. That means they can make the strongest response as quickly as possible. Why have the COVID-19 vaccines worked so well? This is what COVID-19 looks like. You may have seen this before. Firstly, the best part of COVID-19 to target, the bit which makes the strongest immune response, was already known. People have spent the last couple of decades researching vaccines to the SARS epidemic from 2002, which meant that this best target could be used immediately. Secondly, vehicles to deliver this target have been developed for use against the pandemic of either flu or Ebola, which we have been expecting might be the next pandemic. Both flu and Ebola are viruses that are similar to COVID, and so the same tricks can be used against COVID as against those two illnesses. So some of the vaccines have packaged that best target in COVID into a non-harmful virus particle that can't cause an infection, or into a nanoparticle which will look a little bit like a virus. These both look like an invader and get picked up quickly by the sentinel cells. Others have used these RNA vaccines. Now, RNA is a chemical that's a bit like DNA, and all of our cells naturally use it to turn the DNA instructions into active molecules. Here, the vaccines take the RNA instructions for the best target in the virus, and then our cells make it and present it as a template for the immune system. The RNA is very fragile, which is why these vaccines have to be stored very cold, and our bodies degrade it completely and quickly once it's done its job. It would be great if we could use these tricks against the next pandemic after COVID as well. If we look at the worst pandemics in history, these are the top 10. You can see that COVID now is about number 9 and likely to go higher in that. About half of them have been caused by viruses and the other half by bacteria. The technology that's provided our COVID vaccines should be effective against all of these other viruses that have caused pandemics in the past. The bacteria that cause pandemics, these top 5, can all be treated with antibiotics. However, it would be nice if people didn't have to get sick before they're protected from disease. Also, we know that there are some bacteria today that are resistant to all of our current antibiotics. Vaccines can also be made against most bacteria. The best bacterial vaccines use the same trick that we use against the COVID of taking a best protein target, but they also take some of the sugars that coat the outside of a bacteria. And these sugars cannot be targeted using the technology that's working against COVID because it can't make these sugars. So what we want to be able to do to protect against the disease X will depend whether it's a virus or a bacteria. Most likely, it will be similar to one of the diseases that we've already seen. We know that our challenge with the viruses is to speed up the production of getting from knowing what the virus is to getting a vaccine even faster. That would reduce the disruption society faces from the next pandemic. For bacteria, we can use the same technology we've already got to get an immune response. Together with antibiotics, that might be enough. But there are also scientists around the world, like myself, developing technologies to add these sugar coats of bacteria to the vaccines, and hopefully these will be ready in time for the next bacterial pandemic. As well as stopping the next pandemic, there are still infectious diseases today that don't have good vaccines that affect millions of people, like the 4 I've put up here. Maybe we could have a vote on whether we should apply the same message to these diseases that we're applying to COVID, that nobody is truly safe until everyone is safe. Thank you so much for listening. I'll leave you with Anna.
Thank you so much, Dr. Nicholas. I think the last message is super important. No one is safe until all of us are safe. So remember to write down your comments and questions in the chat box, and we will come to them in our Q&A session. So now let me introduce you our second and fascinating speaker, Sophie Hawkins. She is a PhD student at the University of Exeter working on neurodegenerative diseases. And today, her talk will discuss how to use stem cells to model neurodegenerative diseases in a dish. Sophie, the stage is yours.
Hi, good evening, everyone. So as Anna just said, tonight I'm going to give you a talk about that will give you a basic overview of a model that I use in my PhD for investigating a neurodegenerative disease. So the disease that I look at, I'm very sorry here, I'm having some technical difficulties changing my screen. Sorry, could we pop Anna back up and I will go on to the next talk after, please?
Okay, Sophie, don't worry. You can check that. So we can show our lighting talks right now. So I'll introduce you to our first flash talk. We will see a video from inspirational Harriet Earle-Brown. She is a PhD student at the University of Exeter, exploring how the bodies of homeless women are perceived and treated. Today, we will see her talk about the book that she co-authored that exposes the issues faced by homeless women, and a very interesting video. Can you broadcast the Harriet video, please?
Hi, everybody. Thank you for joining me. Today, I'm going to be talking to you about Little Miss and the bodies of homeless women. The bodies of homeless women are often judged and treated badly. This might be because of a lack of access to showers or toilets, maybe leaving the body dirty or not smelling great. It could be because they have fewer clothing options, so can't necessarily dress in the way that women are expected to. And on top of all of this, it can be really hard to keep on top of grooming. So the surface of the body of a homeless woman is often judged and they're treated poorly. But the body of a homeless woman is also incredibly vulnerable, and homeless women are subject to physical, verbal, and sexual abuse. However, the issues that homeless women face are often overlooked and ignored. They're not talked about enough in public policy, in research, and they're not often accounted for in service provision. And so attention needs to be raised to this issue. I created a book which aimed to share the research and knowledge that we have on this important issue in a way that's a little bit more interesting and accessible than a typical academic article or even a standard report. I created Little Miss Homeless, which is a culture-jammed version of the popular children's book. It aims to educate wider public audiences on the issue of women's homelessness, and the book is based entirely on true stories from my own research and that of other people. I collated lots and lots of stories together and picked up the themes that were reoccurring to make sure that this book was representative of many homeless women. This is an example of some of the content in the book. So you can see I've drawn the picture here to look very similar to the original Mr. Men and Little Miss images, and on the left-hand side, I have an extract from the book. It says, "Little Miss Homeless hates having nowhere private to shower, dress, and use the toilet. Sometimes she will go to a homeless charity, but they are only open for a few hours each day. Most of the time she uses public toilets. The sinks aren't very nice, but she doesn't have much choice. However, they get locked at night, so she has nowhere to go to the toilet. This is especially challenging when she has her period. Sometimes she has to change her tampon in a bush in the park." So this is a very real issue that many homeless women face, the issue of being on your period when you're homeless and you don't have somewhere safe or regular to sleep. Little Miss Homeless was predominantly a rough sleeper, but homeless women can be homeless in many different ways. And in all of those ways, having your period is made much worse by being homeless. The reception of the book has been quite well received. I've had over 3,000 digital downloads and 100 physical copies sold. The digital downloads I made sure were free to access, and on top of this I provided my contact details so people could access the PDF if they needed it in staff training. I've had people from the councils, police and mental health wards reach out to me to ask for PDFs and copies of the book so they can use it in staff training. The book has now been disseminated into libraries around Devon, and I've begun sending it into schools as well. And on top of this, I've presented the book at multiple conferences, both academic and non-academic, mostly working with homeless service providers to share this book with a group of people who are able to help. Those who work in the homelessness sector have found this book really helpful, and some of them have started incorporating it into their training just to make sure that homeless women are not overlooked in those conversations. At the back of the book, there's pages like this, and it's very easy ways that the public can help homeless people, and not necessarily homeless women, but homeless people in general. So it's little things like where to donate or what they can donate, as well as how they can engage with different organisations. And I've tried to come up with ideas that match everyone, no matter their age, ability or financial status. So this is hopefully going to make it easier than ever for people to make a difference in the lives of homeless people and homeless women, as well as educating them on this issue. Thank you for listening to my talk. If you'd like a copy of the Little Miss Homeless book, you can access it for free on issue.com, or you can contact me to purchase a physical copy. I've put my social media links down below if you want to keep up to date with the Little Miss Homeless stuff or any of my other research on homelessness and homeless women. Thank you for listening.
Thank you so much, Harriet. That's very inspirational, and the video was very interesting. If you want to contact her, please, you can write directly to her. She won't accompany us in our Q&A session, but it's a very interesting topic to reach after. So I think we are back with Sophie. So please, Sophie, now again, the stage is yours.
Okay. Hello, apologies for those technical difficulties. So tonight I'm going to give you a brief talk that discusses the model that I use in my PhD to investigate a neurodegenerative disease. So the disease that I work on is called amyotrophic lateral sclerosis, or ALS for short. And also you can see the picture here of Professor Stephen Hawking, and some of you might have heard of ALS through association with him. Or perhaps you might have taken part in the Ice Bucket Challenge that went viral a few years ago. But for those of you who haven't heard of ALS before, this is a fatal motor neuron disease, which is characterized by the loss of the nerve cells which connect to your muscles and enable movement. So this is what a motor neuron looks like. And this area here, this is called your cell body or your soma. And these sort of spiky bits that you can see up the top, these are called dendrites. And these dendrites, they receive signal from surrounding neurons. And the motor neuron will pass this signal down along its axon to these areas at the bottom, which will synapse onto muscles and release a chemical signal that tells your muscles to contract. So you can imagine if you lose this cell type, you lose the ability to communicate with your muscles. So for sufferers of ALS, this means that they will gradually become paralyzed as the motor neurons degenerate. So in order for us to be able to come up with an effective treatment for ALS, we need to be able to interrogate the motor neuron directly. So that raises a couple of problems for this, because your motor neurons are so deeply embedded in our anatomy that it's quite impossible for us to isolate these from a living individual. So there are two types of motor neurons in the human body, and the first originates in the brain in a region called the motor cortex, and these then travel down into the spinal cord. And in your spinal cord, they'll then connect onto lower motor neurons, and then this is the neuron which will then travel out of the spinal cord and to your muscle. So if you imagine as you're sat now, all of the different muscle groups that you have in your body. So every time you wiggle your fingers, every time you wiggle your toes, you walk, you talk, there's going to be muscles and motor neurons that are involved in that movement. So if we go back to the structure of a motor neuron, so this cell body, this stays in your spinal cord, and it's this axon which will travel out all the way to your muscles. So if you can imagine when you're wiggling your toe, how long this axon is going to have to be in order to pass that signal all the way down to your feet. So especially for taller individuals, these cells can be over 1 meter in length. So that should highlight to you how we are unable to take these from a human. So as an alternative to that, and due to some really great progress in science over the last few years, we can actually now make the cell type outside of the human body, and that's where the disease in a dish model comes in. So this is the model that I deploy in my PhD, and some groundbreaking work went into the development of these models. So back in the early 2000s, this person called Yamanaka and his group, they worked out that you can actually reprogram skin cells back into a stem cell. And that's really important for us, because if you go back before that point, if you wanted to obtain stem cells from an adult particularly, that was a very painful procedure, and the stem cells that you obtained were already directed towards a certain lineage. And if you wanted a stem cell that could be differentiated into all cell types in the body, you'd have to get that from an embryo. And obviously that threw up quite a lot of ethical concerns from people. But now what we can do is we can take a skin biopsy from an individual with ALS, and you can reprogram those skin cells back into a stem cell, and then you can use that stem cell and differentiate it into a motor neuron. And then we can study that motor neuron in a dish. Now, if you're following the Nobel Prize Awards in the last year, you might have heard of something called CRISPR-Cas9. So 2 ladies won the award in chemistry for their work on this in the last year. So this system is a way for us to edit the genome of cells. So we can now take this patient-derived stem cell, so we've obtained this from an individual with ALS, and we can go in and we can correct that gene that's linked to ALS so that we get another cell line. And when we make neurons from this, the only difference between these neurons is going to be that single gene. So when we're trying to work out what's different between our diseased and our corrected lines, we're removing all of the confusing, confounding variability that you get between individuals. So I thought I'd include a couple of images of what I would see on a day-to-day basis. So these images that you can see here, the cells that look a little bit like a blob, this is what a stem cell culture looks like. And the cells next to it, this is what some cells look like when they're on their way to becoming motor neurons. So once we've made the motor neurons, we have to show that actually we're making what we say we make. So these images are a technique called immunofluorescence. So what we do is we probe neurons for proteins that we know are expressed in that cell type. So this image on the left, what you can see in bright green, this is for a protein called MAP2, and that's going to stain this cell body region and these dendrites. In the pink and the red that you can see in both images, that's staining the nucleus region. And then the turquoise color that you can see on the right, this is staining all of the neurites, so that's going to include this axon as well. So this is a technique that we use to work out that the cell type we're making is indeed motor neurons, and then we can work out the efficiency of those differentiation protocols as well. There's something else we can do. So remember I talked about earlier how these dendrites collect signals. So motor neurons are known for being able to pass signal to muscle. So what we can do, we know the kind of molecule that is usually used to signal to a motor neuron. So we can wash that molecule over the top of our neuron cultures and see if our cells are capable of responding to that. So the video I'm about to play for you is us washing a chemical stimulant over the cells, and we have a dye that's able to capture if the cells respond or not. So that color change that you saw from blue to yellow, it was very quick. That is just telling us that our cells are capable of responding to the stimuli that they should be responding to. And once we have our neurons, we then start comparing our ALS neurons to our control and our healthy neurons, and we start to probe what are the differences between these. So this can be things like looking at gene expression. We can look at the shapes of the neurons. So are some longer or shorter? Do they survive differently? What about, are they able to respond to stimuli the same? Can they pass and receive signals? All that sort of thing. And if we find something interesting that we think we're able to correct, we might try out that treatment in the neurons and see if we can rescue the disease somewhat. And that might be something as basic as just following the survival of the cells to see whether you can improve the survival of the ALS neurons. So I think that's about all I've got time for. So thank you for listening, and I hope you enjoy the rest of the talks tonight.
Thank you so much, Sophie. That was a very interesting talk about neurodegenerative diseases. So remember to post all the questions for Sophie in the chat box. We will come to her in the Q&A session. And remember that we have a question for the audience that you can answer in the chat box during all the presentations. So the question is for you to remember, what is the first thing, activity, travel you would do after receiving the COVID-19 vaccine and you are free to go anywhere? In the Q&A session, we will come to some of the best answers. So it's time for our second lightning talk. Let me introduce you, Dr. Piotr Slowinski. He will give us the second lightning talk. He is a research fellow at the University of Exeter, translating quantitative methods into healthcare technologies. Today, he will show us how augmented reality technology helps in diagnosing brain disease and mental illnesses. Pietro, the stage is yours.
Hello, everyone. There is something in the way we move. We share the experience that we usually can tell a person who it is just from the way that the person is walking or from the posture, even before we see it. We can also quite easily tell how the person is feeling, if it's happy, sad, or angry, from the way it's moving its head, its hands, and again, just walking and moving. And medicine confirms these intuitions. The doctors use movements in the diagnostic battery tests to tell if someone is suffering from the neurological or mental disorders. But it is very difficult to do. It requires training, and especially at the early stages of the disease, when the changes in the body are very small, the changes in the movement are very, very difficult to spot. I use mathematics and modern technologies to address these challenges. I use devices like this, augmented reality goggles. So they are like very, very convenient. They are lightweight. They can be easily put on your head. They have all the sensors that are necessary to record the eye movements or to record the hand movements to help doctors to test this disease. And the tests are like simple computer games when you're, for example, following an object that is like moving in front of you on the screen and that is being displayed. So the advantage of using this portable technology is that it can be used at home, it can be used in clinic, and because the data is only being recorded during very specific activities and during very specific times, it's really respecting the privacy. To analyze this data that is recorded through devices like this, I'm using mathematics. And what mathematics is doing is really helping to translate the similarities that we see with our eyes into precise measurements. I used the methods that I developed to show that such movements and such analysis can be used to help with diagnosing schizophrenia and to help novel therapies for developmental coordination disorder. So to summarize, I would like to say that our movements have quite a lot of information about our health, especially about how our brain is functioning, how our emotions are functioning, that new technologies, which are very, very convenient and rapidly developing, allow us access into this information and allow us to record it in a places that are convenient for the patients and for the people. So again, at home or at the clinic, and that mathematics can really help us to see that the judgments that we are doing in everyday life, the similarities that we see between the movements of the people and how they are moving, can be really translated into something precise that can be very useful for doctors. Thank you very much.
Thank you so much, Dr. Piotr. That was so nice, and a nice example of how artificial intelligence can be very used for good for our societies. Remember to post your questions and comments in the chat box, and we will come to Dr. Piotr in the Q&A session. And now we are going to our last presentation of today by the captivating Serena Sabatini. She's a postdoctoral research in the REACH team at the University of Exeter in the medical school. She is exploring how we can enhance the life of people living with dementia. And today she will talk about how a positive attitude can help with our relationship with dementia and aging. Serena, the stage is yours.
Thank you, Anna. Well, hi everyone. My aim with this talk is to convince you all that having a more positive attitude towards aging is worth it. But first thing first, what do I mean when I talk about attitudes towards aging? Well, to explain this, I'm going to ask you to do a quick exercise. I will need you to visualize an older man or an older woman. And this should be the first picture that comes to your mind when you think about an older person. So it could be a person you know very well, such as a member of your family, a person you barely know, or even someone you don't know, for instance, someone famous. It could even be a generalized image of an older person. So I estimate that by now, you all have someone in your mind. So those of you who have visualized an older person as someone who is physically frail, in need of care, lonely, sad, or with memory problems, would be said to have a negative attitude towards aging and older people. Whereas those of you who have pictured an older person as someone who spends lots of time with friends, with grandchildren, or lots of time pursuing enjoyable hobbies, would be said to have a positive attitude towards aging. Now, if you are one of those people who have visualized an older person in a negative way, please do not feel bad about that. Research is actually telling us that most people have a negative attitude towards aging. But why does this happen? Why most people have a negative attitude towards aging? Well, this is mainly due to our society depicting older age as a negative and undesirable stage of life. For instance, most of the card shops I've been to sell these sorts of cards depicting older people in a negative way. We are rarely surrounded by more positive pictures like the one shown on the screen. However, it's not infrequent for older people to gather together to engage in sports or other hobbies, to pursue degrees, or even to contribute to society through volunteering activities. These pictures tell us that older people can be active, can be happy, and can have fun. It is true that older age can bring many losses, such as a decrease in physical health. But let's be mindful that older age can also bring positive changes. For instance, it can bring increased knowledge, increased wisdom, and also more free time to spend the way we want. So I guess that at this point of the talk, you may be wondering why I'm interested so much in attitudes towards aging. Why do I care about attitudes towards aging? Well, it looks like that changing people's attitudes towards aging may be a strategy for promoting better health in older age. Indeed, those people who grow up having more positive attitudes towards older age end up being healthier older adults. For instance, they tend to maintain better memory, they tend to be happier, and they tend to be physically fit. So why does this happen? Why those people who have a more positive attitude towards aging end up being healthier older adults? Well, it turns out that when people have a negative attitude towards aging, they tend to think that losses are an inevitable aspect of older age. And because of this, they are less likely to engage in health-related behaviors, because health-related behaviors would be seen as pointless. In contrast, when people have more positive attitudes towards aging, they are more likely to engage in a healthy lifestyle. For instance, they are more likely to follow a healthy diet or to do physical activity. And in doing so, they are more likely to grow older with a better health. So my point here is that the way we think about older age shapes our actions, which eventually shape our health. So lots of the research I conduct has to do with the way in which people perceive either their own aging or older people in general. And now I want to briefly tell you about a research study I've conducted. In this study, I was interested in understanding how people with dementia perceive their own aging and whether this has an impact in the way in which they perceive their life, how much happy they are with their life, and how much satisfied they are with their life. So in order to do so, in this study, 1,541 people with dementia were asked to answer a list of questions. And this question aimed to understand how people perceive their own aging and how much satisfied they are with their lives. For instance, participants were asked whether they agree or disagree that as they grow older, they are less useful than before and things keep getting worse. So from this study, I found that those people with dementia who have a more positive attitude toward aging, they are more satisfied with their life compared to those people with dementia who have a more negative attitude towards aging. So it seems like having positive attitudes towards aging are a sort of resource that not only increase one's likelihood of aging more gracefully, but they can also help those people who are aging with an illness, such as dementia, to be slightly more satisfied about their life. So for those of you who are not familiar with dementia, this is a condition that involves significant cognitive decline. For instance, people with dementia can have difficulty speaking or remembering things. And as the pathology progresses, these difficulties become more and more severe. And because of these cognitive difficulties, people with dementia can have can find it hard to conduct even basic daily activities. For instance, they can find it hard to doing a phone call or doing the food shopping. People with dementia are also more likely to experience depression compared to the general population. So, as there is currently no cure for dementia, all we can do is try to find a way of supporting people with dementia to live as better as possible with the condition. Now, the findings of this study tell us that having a more positive attitude towards aging may help people with dementia to feel a little bit better about themselves and about their life. Now, you may rightly think that it can be hard for people with dementia to be positive about their life and about aging. And the challenge for all of us is try to find new ways to support them to live as best as possible. Perhaps one way of doing this, of achieving this, could be promoting more positive images of aging and more positive attitudes towards aging throughout our society. So if the way in which our society depicts older people change, these may end up in older people perceiving themselves in a better way. Thank you all for listening my talk, which was the very last, and I'll leave the scene to Anna.
Thank you so much, Serena. That was a very interesting talk and a message for all of us, because we are all going to that point that we have to be more positive about our aging life. So let's thank all our speakers. And now it's time for our Q&A session. And remember that you can post more questions during this session too, and we will go to there if we have time. If not, you can reach our speakers, and you can come back to the video because it will be posted on YouTube after the live broadcast finishes. So let's talk about some of the answers to our future plans after the COVID vaccine is applied. So we have our first answer from Johan. So he says that the first place that he will go is definitely the pub. So we all agree a bit that in going to any other place, to our places, and here in England, that there are a lot of pubs everywhere, would be so nice to gather again with friends in those public spaces. There is a very nice comment from Kelly Clark. She wants to hug her mom and her nanny again. That will be also awesome. See again all our relatives, on all our more older relatives that are on risk. So that will be incredible, awesome too. Okay, thank you so much. Now I will invite our speakers to join us in the Q&A session, and I will start to some questions for you. So there is a first question for Nicholas from Paula. So the question says, do we have a problem with overuse of antibiotics?
We do have a problem with overuse of antibiotics, yeah. The problem is that when you use antibiotics, you select for bacteria that have got some way of resisting the antibiotics. And those bacteria then will survive, and they very often then go on to infect somebody else. So what we need to do is to use antibiotics more sensibly and only use them when it's necessary. So I think that the problems we tend to find are when they're used, for example, in food production, just to make animals produce food a bit quicker because they don't get minor infections, then that's a problem because it causes resistance. Similarly, in humans, quite often people try to get antibiotics when they've got a virus or something like that, and it's not going to make them any better at all, but it does cause extra resistance. The other issue we have as humans is that when we take antibiotics, it also tends to kill many of the friendly bacteria we have in our guts and on our skin, and so it can cause us health problems if we take antibiotics inappropriately that we don't need. So I think in general, what we do need new antibiotics, because there are some bacteria that are naturally very resistant in the first place, but also we need to be much more sensible about how we use them, and to let the medical professionals hopefully make the right choices and not put pressure on people to give antibiotics that aren't needed.
Thank you so much, Nicholas. That's very interesting and helpful for all of us. I have another question for you, because I think it's something that we all are interested in right now. So Mateo says, I would like to know more about how development of COVID-19 vaccines can keep up with all the new variants that keep popping up. So how can be resistant to other variants?
So far, thankfully, the variants that are most infectious are not, so don't seem to have resistance to the vaccine. However, what's likely to happen is that the variants that we're seeing have got very small changes, which are possibly going to cause some problems with the vaccine. The issue is that the platforms we've already got that can be rapidly changed to make a new vaccine, because they're designed to take anything which any part of the protein. So as the virus mutates, what we'll tend to see is that a new vaccine will get made that is going to take the mutated virus rather than the original virus and use that as a vaccine. The trouble is that it does take a little while to make the new vaccines and then get them properly tested and then into production. And that's what the companies who are doing it, they're at the same time trying to ramp up the production of the first generation, and at the same time cut their lead time to get the second generation and make sure that they can put those into production. I think the strength they've got is they've made these platforms so that they've not just got a one shot that will work and then they haven't got anything else. They've got a platform so that whatever changes happen, they can rapidly redevelop what they've got to meet that change. The challenge really for them is being able to get the production up so that we can vaccinate everybody in the world really as quickly as possible. I think we've lost Anna. Okay. Okay.
So maybe in the meantime, while we are waiting for Anna, I can answer some questions about the AR goggles. So yes, first of all, they need to be like tuned specifically. Okay, first about the cost. So the cost can vary. So they can cost from like a couple of hundreds up to like a couple of £1,000. But the one that I envisage as being like being useful at home will be like, in the cost of a couple of £100s, and this is like much cheaper than any kind of EEG imaging machines, or like much bigger setups that require, like, for example, a full room in the GP's office. So then yes, there's questions. Yeah, so handwriting is one of the fine motor skills, and I heard about the studies. It's being used, for example, to also help to diagnose schizophrenia. And there are differences in how people are signing their name and how they are using the pen, and the fine motor skills can be even used to try to tell something about side effects of drugs. And in this case, such an activity as handwriting would be translated into some other kind of movement that is happening in front of the eye. So information that is being extracted from the taking the signature or handwriting can be also extracted from the games that I'm developing. And going with the next one, Yes. So each of these games, so there will be like a couple of activities. So in some game, you will be picking like objects that are in 3D in front of you. In some of them, you are going to follow like some very, very precise movements on the skin. And in others, it will be combination of interaction with couple of virtual agents that are going to be displayed on the screen. And then depending on the condition, different aspects of movements that are being recorded in the settings can be used to try to help with diagnosis of different different conditions. So to some extent, the calibration in a traditional way, in meaning that you have to do some very, very precise movement first to set up like some initial measures on the movements, are not necessary when using these setups. They're being done in the software beforehand, but the tasks and the measures extracted from the task will be tuned to some specific conditions.
Thank you so much, Piotr. So there are some questions for Serena.
Okay.
So the first question would be how to measure that positive attitude from Matteo.
Yes, well, the way we measure it, we ask people some questions. So they read some questions that we call items, and they just answer whether they agree or disagree. For example, we ask people whether they think that as they get older, they are less useful than before, or whether they think that as they get older, nothing has changed in their life. So we ask people basically whether they have experienced some changes or not, and then depending on their answers on scores, we can see whether people have a more positive or a more negative attitude towards aging.
Okay, very interesting. Thank you so much. Another question for you, and I think also if Sophie wants to come up after Serena, because do you think a positive attitude would help to improve the brain at neurological level?
Well, yeah, there's some research showing a link between the attitudes that people have towards aging and their cognitive health. However, it's not something, let's say, if you change your attitude when you are 50 or 60 or 70, it's not that suddenly your cognition will change. Those people who grow up since early ages having a more positive attitude towards aging tend to experience less stress, tend to follow a healthier lifestyle, and all these factors increase your likelihood of maintaining cognitive health in older age. So there's more about that. It's more about your lifestyle. But what's very important is that since when you are young, if you think that older age can be positive, I guess that you are more likely to do something and make it more likely for yourself to experience better health in older age.
Okay,
so I just wanted to say that we are all happy to be contacted, and we are all happy for all of you to write down questions in YouTube, and we will try all to answer, because I guess there are quite a few questions.
Yes, there are more questions. I have a question for Sophie that comes with the question of Serena. Do you have experiments with different cells with different people ages? So
yes, so the stem cells that we have, we tend to have the different ALS mutations. So there are lots of different mutations that are linked to ALS. So those will be from people of all different ages, different sexes, different ethnicities. So generally, when we try and keep controls, we try and match them somewhat. So if we have a healthy control that hasn't been gene corrected, for instance, if we have a female iPSC line, we will try and match that with a female donor of roughly the same age. So yes, we do have variation in that, but ALS is generally sort of an older disease that most people that develop it will be sort of middle-aged and older.
Okay. Thank you so much. So I think we have a lot of questions now. So as Serena mentioned, we can you can reach them. Also, there is an interesting question for Harriet that I will let it open for people that want to answer, and also if Harriet herself can answer later. So what needs to be done by the government, local authorities, to really help the homeless in the UK? And like has a big question for all of us to help them to feel better and safer also. So I think that we are coming to our end. Please remember that the show stays on YouTube after the live broadcast. It is time to thank and clap to all our amazing speakers and to all of you, our fantastic audience, for your time and your participation. Thank you so much for joining us. And I want also to thank all the members of the organizing team. So to our producers, Valentin and Alex, to our Master of Comments, Catherine, our YouTube administrator, Lydia, and finally to the lead of all these sessions, Serena. Thank you so much for all the work. And the other organizers, and I hope you have enjoyed the session and learned a lot like us, and that you now have more questions for the future about health. Have a good evening, stay safe, and remember that this is the first day of the show, and don't miss other sessions of the Pines of Science too. Cheers.
Cheers.
Heard about PCR biosystems?
There's quite a buzz about us.
Hop on over to our website to find out more.